These are your video notes for fossils, a brief timeline of evolutionary history, and some experiments on the origins of life. Let's go ahead and jump in. So, one of the main lines of evidence that particularly Darwin relied on and that we still very much rely on today is fossil evidence. Since our discovery of DNA and gene sequencing, DNA evidence has kind of become the primary line of evidence for evolution, but fossils still play a major role and are incredibly interesting in their own right because they provide a history of life on Earth, right? A fossil is just any remnant of an organism, right? It could be like fossil bone, fossil teeth, could be fossil footprints, right? And we use these fossils to examine kind of the natural documentation of how organisms have changed over time, because change in structure can get reflected in these fossils, and then we compare the structure or the shape or the style of fossil to perhaps a current modern version of that same species, and we can just see how organisms have changed or how species have evolved. Sometimes we discover that many species go extinct, and we've actually realized that this is the main life cycle of species. Just like an individual might be born, grow, and then die, it's natural for species to evolve over millions of years and then eventually go extinct. And actually, the average lifespan of a species is about 10 million years on average before any given species goes extinct. Humans are only about 250,000 years old, so we're a very, very young species. We're a fraction of the way to being average, but we do see that many fossils just stop, either in number or just completely just don't show up after a certain point. And species go extinct. Paleontology is the study of fossils. You're probably also a little bit of a geologist because those two are inextricably linked together, and we're going to see that here. So again, fossils are any evidence of an organism. It could be a complete organism. It's rare, but if it happens, it's usually something small or some of these more primitive kind of single-celled eukaryotes with your protists or your prokaryotic bacteria. Usually it's part of an organism, but it could also be footprints, eggs fossilized, seeds and pollen fossilized. You could even get poop fossilized, known as coprolites. But again, all of these give us a peer or a look into the past to see the structure, the shape, the size of different organisms and what they looked like potentially tens, hundreds, or in some cases with bacteria, billions of years ago. So the main way fossils form is that the atoms that build those teeth or those bones are very quickly buried by sediment. And this is one of the key features for becoming a fossil. Quick burial is one of the primary ways to become a fossil. And we're actually going to list these out. So quick burial is number one. Number two is that you are hard-bodied. You don't have to be hard-bodied, but it certainly helps if you're, you're not all skin and muscle, and if you're all soft tissue, that's probably going to decay away. It's the hard tissue like teeth or bones that are much more likely to fossilize. And the third criteria that I want to talk about is low oxygen. It's usually in your benefit to be buried quickly in a low oxygen environment because oxygen and kind of the metabolic processes, aerobic respiration that use it contribute to decay. So quickly buried, hard-bodied or hard-bodied components in a low oxygen environment. Best chance to become a fossil. Typically, this means that most organisms that are fossils are usually coastline or aquatic species, and that's another key thing. You're usually aquatic or you live near the coast, because moving water is an excellent way to carry sand, dust, mud, etc. that can bury you. Right? So that sediment, that piling on the sediment is a key feature. Much more difficult to decay if you're far inland. Most fossils are found in a specific type of rock called sedimentary rock. That's the kind of rock that as layers of sediment and dirt and mud and silt compress over time, they become compacted. Shale is a good type of rock that falls into this category. It's also the type of rock that we use, that we drill through to get like natural gas, right? Sandstone is another type of rock that falls into this category. Slate is another type, right? These are all types of sedimentary rock. Because material gets deposited on the surface of Earth and compacted over time, older layers of rock are towards the bottom. So if you're looking at a canyon or a cliff face, like the side of a mountain, the lower rock layers are the older layers, and the newer layers are the layers on top. And this is useful because if we have fossils in layers higher up, we know that they are younger than fossils found in layers closer to the bottom. So, quick recap. How to become a fossil? Again, usually being near water, beneficial because water carries sediment. That sediment piles up on top of you, and basically, atom for atom, the minerals that comprise your tissue are replaced by stone or sediment that hardens into stone or sedimentary rock. So really, when you're looking at a fossil, it's not bone anymore at that point, or it's not teeth. It's actually rock. And then hopefully we find these, realize that they're not just regular old rocks, and we study them and categorize them and use them to update the fossil record. And the fact that older fossils are at the bottom allows us to start applying some times and dates to the fossils that we find. This is called relative dating, and it's one of two major ways to date or determine the age of a fossil. By dating, I don't mean taking it out to a restaurant and, buying a fancy dinner. I mean like figuring out how old it is, right? Higher rocks, kind of off the surface of the earth, contain younger fossils, and the rocks themselves are also younger. So if you have, let's look at this fossil right here is going to be younger than this fossil down here, right? And that's really all you can figure out with relative dating, more or less. There are things called index fossils, which is a fossil of a species that existed worldwide or had a very wide geographic range and existed for a short amount of time. And the point of an index fossil is to basically use a reference. It's like having a ruler and measuring something. Worldwide distribution is important, or a widespread distribution is important because it allows everyone across the globe to use the information, just regardless of where you live. And knowing that that fossil existed for a short time allows you to pinpoint a specific date for that fossil. So let's say you have a fossil right here, right? In this layer of rock that you know existed for only 5 million years worldwide. And you know that that fossil, via a dating method that we're going to talk about very shortly, is 510 million years old. Well, if you have a fossil just beneath that, you can say, well, hey, this fossil that I discovered in this kind of red layer right here is right below my index fossil that I know the date of. So this fossil must be slightly older than 510 million years, maybe 520 or 530 million years. The second way to date a fossil, which is much more accurate, is radioactive or radiometric dating. This is where you use isotopes. Remember that an isotope is a radioactive or unstable version of another atom. And you can treat them like a clock. They're going to decay. And as they decay over time, you can measure the amount of that unstable isotope that remains. And the half-life is a unit of measurement that tells us how long it takes half of that unstable isotope to decay. And the one that we're going to do a sample calculation with is carbon-14. So if you guys remember from maybe some chemistry or physical science, carbon has an atomic number of 12. If you add all of the protons, the positively charged units, with all of the neutrons, the neutral charged units, in the nucleus of a carbon atom, you add those all up, you're going to get 12 of them. However, there is carbon that contains two additional, that's supposed to say plus two additional neutrons, and its atomic number is carbon-14. That's the unstable version, and it will decay. And half of it will decay every 5,730 years on average. And we can use this information to predict the age or measure the age of a fossil, because after one of those half-lives, so this is when the fossil, or sorry, this is when the organism has died at time 0, right? One half-life later, so 5,730 years later, half of our carbon-14 has decayed. 50% is remaining. Another half-life passes, so another 5,730 years pass, and we're now at 25% of our isotope remaining, and so on, and so on, and so on. And we can use this to predict or measure the age of a fossil. So here's an example. Let's say you're an archaeologist and you have dug up a fossil or an artifact and you want to know how old it is. And you have been able to determine, and there's an excellent video that I will show you in class. I'm not going to put it in here because this is just a lecture and a sample calculation. But, you measured the amount of carbon-14, the unstable isotope, and there is 12.5% of the original carbon-14 content remaining in that fossil or that artifact. How old is it? Right? How old is it? I would recommend doing a T-chart like this. So on the left, we're going to have the percentage remaining carbon-14. How much carbon-14 is left, right? And on the right, we're going to have the years, the time. Right. So on the left, you have 100% of your carbon-14 remaining at time of death, right? After one half-life, you're going to have 50% of that carbon-14 remaining. And we're just going to keep halving it. Half of that is 25, half of that is 12.5, and actually that's the value that we are most interested in here. So 0 years have passed, all of your carbon-14 are present, right? After one half-life, from 100 to 50%, 5,730 years have passed. After a second half-life, when only 25% of your carbon-14 remain, another 5,730 years have passed. After a third half-life, when you've gone from 25 to 12.5% remaining of carbon-14, another 5,730 years have passed. So how many years have passed? How old is this artifact or this fossil? Well, add your years up. What is this, 17,000? Should have a calculator. Hold on, I'm going to pause the video. All right, I'm back. 17,190 years. 190 years. So it's almost certainly not going to be, it's not going to be like a dinosaur species. You'd have to go back like 70 plus million years for that. But this very well might be like early human ancestor before we settle down for agriculture, right? Agriculture is about 10,000 years old, so maybe this artifact or fossil is from a nomadic human tribe, potentially, or just a completely different species altogether. But you do need to know how to do this type of calculation. Very quick summary of the history of life on Earth. Earth is 4.5 billion years old. It's an old planet. And we figured this out using a couple different dating methods. The one that is most precise, I believe, is the dating of zirconium crystals, which there is a good Cosmos video of with Neil deGrasse Tyson narrating that kind of describes how that process went about and how they figured it out. And I would definitely do a Google search or some research and check that video out. But we've also learned that Earth's early atmosphere was actually incredibly hostile. Carbon dioxide gas, carbon monoxide, nitrogen, water vapor, hydrogen cyanide, hydrogen sulfide, these are not gases that are necessarily conducive to early life. They were a very toxic atmosphere. Something that Alexander Oparin referred to as reducing atmosphere. A reducing atmosphere, which is something that you might encounter, and I just want you to be familiar with. Oparin is the scientist, and the reducing atmosphere is basically referring to the type of chemistry that these gases participated in. But the point is that when you're forming a rocky planet, I mean, rocky planets are very hot when they're first forming. They're not really conducive to life. But as Earth cooled and liquid or water vapor condensed into liquid, and we had oceans, we had a substrate. We had an environment for life to start to evolve on. And our oldest evidence of life is 3.8 billion years ago of mats of prokaryotic cells that built up in these awesome looking structures called stromatolites. Stromatolites. And stromatolites are our oldest fossils of bacteria in an environment when there was no oxygen. I'm going to show you a picture of what stromatolites look like. And this is just a quick Google search. Google stromatolites. They're these kind of, I don't know, a good way. They look like little rock structures kind of coming out of shallow bodies of water. But they are our oldest living life forms. They are the oldest fossils we have, and we've dated the rock and the surrounding rock. They are 3.8 billion years old. So it took, if you're just kind of doing some back of the envelope math calculations here, it took about, let's see, what, 1.5-ish billion years. Oh, I'm looking at the wrong number here. Yeah, over 1 billion years for Earth's early chemistry to form, the chemistry of single-celled prokaryotic cell. That's a long time. I mean, humans live, what, 80 years on average, if all goes well? I don't know, our brains are small. I think it's difficult to figure out and appreciate the time span of 1 billion years, but that's how long some of the first cellular chemistry took to evolve. And then it took over another 1 billion years to get us photosynthesis. We're still with prokaryotes here, we're still single-celled, but now we have photosynthesis, right? Photosystems 1, photosystems 2, chlorophyll, the production of oxygen. Well, how do we know? This is a good question to always ask. How do you know when photosynthesis happened? We surely were not around over 2 billion years ago, 3 billion years ago. Like, how do we know this stuff? And the reason we know that photosynthesis first evolved about 2 billion years ago is because oxygen gas rusts rock containing iron. So what we've done is we've gone back and we've looked at iron rock. I'm going to go back out of the PowerPoint for a second. And this is a beautiful example of a rock formation in Australia. These rocks are red because they contain iron, and they're only going to rust or oxidize in the presence of oxygen. And the fact that they are red means that when these rocks formed, oxygen was present in the atmosphere. So we went back and we dated, using those techniques that we talked about, the rock. We figured out how old the rock was, and the rock was over 2 billion years old. And we've found this data to be supported on every continent with every kind of rock that has iron. Oxygen originated in this atmosphere over 2 billion years ago as a result of the only process that could have put it there, which is photosynthesis. So therefore, we know that photosynthesis evolved over 2 billion years ago. I think that's super cool. What else? We've figured out that eukaryotic cells evolved about 2 billion years ago, invertebrates at 700 million, and vertebrates, starting with your fish phyla and your fish groups, at about 500 million years ago. All right, one quick discussion about the origin of life. So, can you get living material from non-living material? In other words, can non-life be the substrate for life? And the answer is absolutely. Yes, this is called abiotic synthesis, and it was confirmed in an experiment from Miller and Urey and has been subsequently repeated. And it's one of my favorite experiments. They tried to create some precursors to life, or they basically just set up a beautiful contraption that mimicked Earth's early atmosphere. Hydrogen, methane, ammonia, ammonium, water, and put that in a contraption that they heated. And they heated it up because, as rocky planets form and the friction from colliding rock generates heat, rocky planet formation is very violent and it's hot. So this would be a heated kind of chamber with all of these gases in it that we know were in Earth's early atmosphere. And as this was heated and this water vapor and its contents rose as hot gas gets hotter, it expands and rises. So that's what this ascending tube is for. And then as that water vapor and contents cools, it passes through a second chamber up here in the top right that had an electrical spark, literal electricity running through it. And that was to mimic the presence of nitrogen. So we started with basically a model of Earth's early ocean. We're passing through Earth's early atmosphere with lightning discharge or electrode discharge simulating lightning. And then as this continued to further cool and condense and precipitate, basically just like it rains now when we have precipitation, all the way down into this bottom collecting duct. And what they found is that this duct, after just a couple days, started to turn like this dark purpley black color. And it didn't start that color. It started, clear like water would look like. And it ended up this dark purple color, and they assayed it. They did a chemical assay. I probably spelled that wrong. But they figured out that they actually had produced all of the precursors to all of our macromolecules. They produced amino acids, the precursors to proteins, hydrocarbon chains, the precursors to lipids, nitrogenous bases, the precursors to DNA and RNA, simple carbohydrates, the precursors to larger polysaccharides. It was possible, in fact, very possible, likely given Earth's early conditions, that non-living material could easily develop into the precursors that we know life needs on this planet. And that was the beauty, the absolute insight to this experiment. And this is why I love this experiment so much. Here's an example of the actual lab setup. Again, think of the creativity it takes to develop this kind of experiment. Never let anyone tell you science is boring or not interesting, right? Here it is again, University of Chicago. I love it. I love this experiment. I'm going to end this video with the endosymbiotic theory, or the explanation of two eukaryotic structures in particular. This is something we have talked about, but I will bring it up one more time. Remember, Lynn Margulis was a woman in STEM, one of our main female scientists, Marie Curie and others being some of the other prominent female scientists. I always like to point them out. It's not all men doing this. More women in STEM is the way to go. First, eukaryotic cells formed by prokaryotic cells living inside of other prokaryotic cells. Basically, you had an aerobic bacteria that was engulfed by a larger cell but not digested. It retained its function and is the ancestor of all modern day mitochondria. The same thing is true for chloroplasts. In this case, the bacteria had photosynthetic abilities, was not digested, and became incorporated into that larger cell. This has been confirmed because the evidence for it is ridiculously overwhelming. How do we know that this is what happened? Well, we know because these structures, mitochondria and chloroplasts, have their own DNA. They have their own DNA. And it's circular. Well, what are the only living things on this planet with circular DNA? Like literally, their DNA is in a circle, not linear like our chromosomes. Their chromosome is one continuous circle. Prokaryotes have circular DNA. They have their own ribosomes, and those ribosomes are the same size and composition as bacterial ribosomes. The double membrane of these structures betrays the fact that there was an original cell with its own membrane engulfed by its host cell, therefore getting the two membranes. And finally, they are the same size as prokaryotes. Awesome. I hope this helped. We'll talk more about it in class. I will see you then.